All right, so this is what I'm calling miscellaneous translation things, because it's just kind of some other things that are important for translation, but it's not a part of the process of translation. And the first is ribosome, structure and function. We know that they're related because it always is. And I'm telling you now, somewhere in one of your free responses, you're going to be able to talk about how structure and function are linked. I don't know where, but you can put it in there and it'll earn you a point somewhere. Every year my students have said, I've been able to put it in somewhere. And when I read over the questions, I'm like, yep, that's where it could have gone. So anyway, when we look at ribosomes, they are made of two parts, a large subunit and a small subunit. And side note, this is one of the ways we can tell differences in prokaryotes and eukaryotes. Their large and small subunits are different sizes than the eukaryotic large and small subunits. Interesting fact. Okay. So the structure is that they only come together when they're working. There is a location for the mRNA to bind. That's important because it does have to bind to mRNA. And then there are those three active sites where the arriving amino acid is, the one that's building the polypeptide or the protein, and the exit site. And so those are the three locations that are kind of really involved in the large subunit, really involved. The small subunit's job really is holding on to the mRNA and making sure that that the ribosome is moving down the RNA at the right speed. So the structure of the RNA is that, of the ribosome is that. The function is to hold everything together and to help create this bond. So it actually does help form this bond. It has a little bit of catalytic energy, activity, I don't know what you want to call it. So not only does it hold everything in place, so not only does it have a structural job to hold everything there, to hold the structure in place, it also has a chemical reaction or a catalytic job. It's going to do a chemical reaction, which is to form that bond and break that bond. So that's the function of the ribosome. It's the site of translation. The next thing is I can describe the function of chaperonin. So once proteins are made, just like when we made mRNA, the mRNA is not done. It was what we called pre-mRNA, and it had to go through RNA processing. Proteins also have to go through a sort of processing, and one of those things is to fold it into the right structure, because when it comes off of the ribosome, it's just a chain of amino acids. Now those will automatically, because of the R groups, they will automatically start to do some folding, but some of that folding, they need a little help. And so that's where chaperonins come in. They are going to chaperone the folding of the protein so that it gets folded correctly, because if it doesn't have the right structure, it's not going to be able to do the correct function. And so it's a super important job because proteins pretty much run the world. We need them folded correctly, and so chaperonins are the ones that do that. And they're just another protein. They look like a little canister. The protein goes in, the new protein goes in, a little magic happens, the cap pops off, and out comes the folded protein. The next thing is I can transcribe and translate let me get this there, a section of DNA. Now you did this in an activity, hopefully, if you did that activity. I'm not going to go to traits because we don't have all that with me, but basically what you want to remember is when you're going from DNA to RNA, T goes to A goes to U because there is no T in DNA, and then G and C are still together, so they go back and forth. So wherever we have a T, we're going to put an A. Wherever we have an A, we're going to put a U. And wherever we have, that should have been a C, because I want this to be the start codon. Wherever there's a C, you'll have a G. Okay? Then A goes to U, C goes to G, A goes to U. TTT is going to be AAA. C goes to G, G goes to C, A goes to U, A goes to U, C goes to G, and T goes to A. Okay? Now, most proteins are hundreds of codons long, hundreds of amino acids long, minus 5. So, obviously, it's a little different. Then what I'm going to do, and I'm going to practice this with the chart and the wheel, so you'll have exposure to both, is I'm going to do the first two here, and then I'll pull the wheel up. So the AUG, the first base is A, the second base is U, so I know I'm going to be in this box somewhere, and then my third base is G. Well, there's 3 G's to choose from, or 4 G's, I'm sorry, and I want to make sure that I'm at the correct G. So I need to be at the G that's across from my A. Okay? So the first base tells you what row, the second base tells you what column, that puts you in a box, and then the third base tells you which one of the 4 in that box. And so that is methionine. UGU, UGU is cysteine. All right, let's switch gears really quick and look at the codon wheel, just so you've been exposed to it as well. AAA, first base is in the center, second base is in the middle, and third base is on the outside. So AAA takes us to lysine. GCU takes us to alanine. And UGA is a stop codon. I don't know why they have a black box there, but that's what that means. It's a stop codon. And so that's all you need to do is you need to remember the base pairing rules to get from DNA to RNA, and then you need to know how to read either the codon wheel or the chart in order to get your amino acids. And the last two are I can explain what it means to say that the genetic code is universal and it's redundant. So the first is universal. What we mean when we say the genetic code is universal is we are saying that all living creatures that we know of use it. So if I study a bat, let's say I'm studying bats, and I find some of their DNA is AAA, that's going to transcribe into UUU, and that is going to be phenylalanine. It's going to do that in me, it's going to do that in the bat, it's going to do that in a bacteria, a pig, a pine tree, a whale, a jellyfish, literally everything. There are a few exceptions due to mutations, but the vast majority of things, and even the ones that have exceptions, the vast majority of their genes are still using this universal code. There's just a few exceptions because of mutations. So that's what it means to be universal. It's also why we can take a gene out of a human, like the insulin gene, and we can put it in bacteria and it makes insulin. Because the gene for insulin is a certain sequence, A's, T's, C's, and G's in DNA, that gets translated or transcribed into mRNA by the bacteria using the same code, T to A, A to U, G to C, and C to G. And then they use this same code to get from the mRNA to the amino acids. So when I put that insulin gene, those instructions for insulin in bacteria, they do transcription and translation and produce the same thing that we produce because they use the same code. So that's the universality of DNA. It's universal. The other is the code is redundant. And to be redundant means to repeat. If you say, I can't think of something right now, of course. Like if you say the MCAT test, okay, that's the test you take to get into medical school. Well, the MCAT stands for the Medical College Admissions Test. So if you say the MCAT test, you're saying the Medical College Admissions Test. That's redundant because you're saying test twice. The redundancy here is where what we talked about, I think in the other video, about how serine, there are 6 different codons that code for serine. So UC anything, UCU, UCC, UCA, UCG, as well as AGU and AGC will all give you serine. So there are some different codons that bring you the same base. So if you had those different codons are lined up, you would just see serine, 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 serine. And that just means that there's some redundancy there. There's some repeatedness there. Alanine is repeated, arginine is a 6, glycine is a 4, leucine is a 6, proline's a 3, threonine's a 3, valaine's a 3, isoleucine's a 4. Proline, threonine, and valine are 4s. Isoleucine's a 3, asparagine and lysine, no, lysine I thought was a 6, maybe not, maybe it's a 2. Asparagine's a 2, aspartic acid is a 2, but anyway, all of them, I think all of them except tryptophan and methionine, I believe, are the only 2 singletons. So all of them have multiple codons that can code for the same amino acid, and that is being redundant. And so that's the redundancy of the code. So I hope that helps. If you have any questions about these miscellaneous things dealing with translation, let me know.